Hey, so today we're going to tackle proteins. So this is our second round of biochemistry. You had nucleic acids a couple weeks ago. This is very important for understanding how proteins work. And proteins are the workhorses of the cell. They do all the work, from sending messages to just being structurally important, to lowering the activation energy of different reactions. Very important for our cells. And so we need to understand really how they work. So the first thing you have to understand is, what are they made up of? And so I brought my little pictures today because I've been enjoying my little paper cutouts. So this is the basic structure of an amino acid. Okay, fun stuff. Okay, so we have a carboxyl group over here. So this is the carboxyl group. And a carboxyl group is made up of carbon, an oxygen, hydrogen, two oxygens, hydrogen, COOH, carboxyl. Okay, on this side, we have an amine group. Okay, and that is nitrogen and two hydrogens. We have carbon base in the middle. We have a hydrogen that sticks off the carbon. And then this is the exciting part. This is the side chain, or more commonly called the R group. Okay, so that is an amino acid. Yay. So this is all very exciting. I'm sure you are at home thinking this is the best day of your life, that you get to learn this stuff. But what we need to figure out is how these things work. So we've already gone through nucleic acids, and we've gone through how DNA is transcribed into mRNA. mRNA is processed if it's a eukaryote. If it's a prokaryote, it's not. And then it's at the ribosome, and the ribosome, which is made of rRNA, is reading it. The tRNA are bringing the amino acids, this thing right here, to the party, and they are creating a beautiful protein that is going to be lovely folded. We're going to talk about that today. Okay, so first thing we have to do is look at protein structure, and then that's really all we're doing today. And we might talk about mutations if we have time, but you can figure that out on your own too. So here is, first of all, we're going to start with the different structures that we have of proteins. So proteins have 4 different levels of structure that they can be. Hang on a second. Let me fix this. I feel like I'm always fixing that. Sorry. Proteins have different levels of structure that they can be involved in, and they all have a primary structure. So primary structure is simply when you have 2 amino acids that are connected together. Okay. Through dehydration synthesis, they form a peptide bond. So it's nice because these are called polypeptides. And so polypeptide is just a chain of amino acids that are all connected together in a long line. And so a polypeptide is made with peptide bonds. That's pretty easy to remember. But if you look right here, I'm going to mess up my paper. That's going to make me sad. H2O, right? So we have a dehydration synthesis reaction. So dehydration synthesis, where we are losing, can't talk and write at the same time, where we are losing a water. Right here, it's going to go poop, drop out, and your nitrogen is going to connect to your carbon. And that's going to make a peptide bond. Okay, that leaves a hydrogen free here, and an oxygen that is double bonded right now free here, although we'll see how that is not double bonded later. Okay, okay, so then we would have another one come along. Do it again. Cha-ching. Until you see what we're getting here. We're getting a chain of amino acids. This happens in the ribosome when the mRNA is coded, and the tRNA brings the correct one of these guys in, and they peptide bond them together. Now, what's the difference? Okay, the difference between these 3. So these 3 are just like a generic amino acid. So it's just generic. What is different about them, if they were different amino acids from each other, like glycine, leucine, glutamic acid, things like that, is the R group would be different for each amino acid. And they would have their own properties of their R group. In fact, here is a chart where you can see 20 amino acids and their different side chains, or R groups. And those different R groups provide a variance in how these amino acids bond together, not their primary structure. Their primary structure is just attaching carboxyl group to amine group. And so not anything to do with that side chain. The side chain, or the R group, sticks off to the side, and it's kind of out of the way on when you're doing your primary structure. And so depending on then what the R group is, the different amino acids will be to have different properties. And that's really important for proteins. So proteins, 100%, it is what structure they have equals what function they do. They will have an active site for a substrate to fit in and lock in. They will have, they'll have like maybe a pocket where they can hold iron if they're hemoglobin. They may have a very specific structure that helps them to support a cell so that they can be like a structural support. They may be shaped into a protein channel that goes through the membrane that only allows certain molecules in, but their shape is very, very important. If their shape is off any bit, it changes the function of the protein, because what they do is based on the fact that something can lock in to the molecule. And if something can't lock into the molecule because the shape is changed, the protein, it loses its function or becomes less functional. And so that's super important. And where that structure comes from a lot is these R groups. Now, you haven't seen that yet. So far, we've just seen the primary structure where they all are lined up in a little row. And that doesn't have anything to do with the R group at all. This very first one is just a peptide bond where you have dehydration synthesis that brings the amino group to the carboxyl group, bonds it together, and makes a chain. So far, that's all we've got. That's primary structure, okay? Secondary structure, however, starts to make some changes. So secondary structure, whenever we start doing that, we are now going to see that we have an oxygen here that has 2 electrons that are available. It's bonded to carbon now, so it's 1. And then you will see that the nitrogen here, or wherever, one of these nitrogens that's in the amine group, the nitrogen has a free hydrogen that's just sticking off, just dying to be bonded to something, okay? And so what happens then is you will be able to see, like this animation here, that when that hydrogen and that oxygen can get close enough together, you will form a hydrogen bond. And that is between the oxygen and the nitrogen. It doesn't have anything to do with the R groups. And it is, it's just a hydrogen bond forming between, still between the backbone of the amino acids, okay? The backbone where they're all the same, okay? Now you can have, depending on how they fold, you can have an alpha helix or a beta pleated sheet, and that will provide a little bit of shape inside the protein once we're finished. Now the R groups are still just sticking out to the side. The R groups are not involved in this at all so far. Okay, so far primary structure is peptide bonds, and they are just bonding the amino acids into a long peptide chain. Secondary structure is hydrogen bonds in between the nitrogen and the hydrogen there, forming an alpha helix or a beta pleated sheet. And then finally, we get to our tertiary structure. Now, this is the big one, okay? So whenever you're talking about, because so far, all of our amino acids would still bond together no matter what, because it's just this base area here, and not the R group, and that's identical. So it wouldn't matter if it was lysine or glutamine or methionine or any of those. It wouldn't matter, because all of the amine groups and carboxyl groups and amine groups and carboxyl groups, they're all the same. And so it hasn't, primary and secondary structure don't have anything to do with mutations. Whenever you have a mutation in your DNA, it affects the structure of your protein. And when it does that, or it can affect the structure of your protein. When it does that, it's because you have a change in your R group. And what's happened is, instead of the mRNA calling for a particular TNA with the right amino acid, now you have an mRNA calling for the wrong amino acid, and that's going to affect the tertiary structure. So here's how the tertiary structure works. In the tertiary structure, your R groups, your side chains, have different properties. And some of them have a positive charge and some of them have a negative charge. Of course, those will be attracted to each other with an ionic bond. Positive plus negative is going to get you an ionic bond. It's going to bond those two together. And so it may be that it folds into this beautiful shape because of you have a negative and positive attraction with the protein. Or some of your amino acids are hydrophobic. They do not like water. And so it depends on where you are. If you're in the cytoplasm, of course, that's like mostly water. If you're in the extracellular fluid, it's mostly water. And so if they're in that environment, then those amino acids that are afraid of water, water-fearing, phobic of water, will tuck inside the protein so that they won't contact water. It's like repelled by the water, and they'll get inside the protein. And so now you're getting some interaction, some ionic bonding with your positives and your negatives. You're getting a folding inside of your hydrophobic parts. And then there's other that are hydrophilic that just love water. And so they'll press out to the outside to try to kind of contact water, unless you're in a no water environment, a non-aqueous environment, such as the inside of the cell membrane. The inside of the cell membrane is lipid, and there's no water in there. And so if the protein is spanning the membrane, you actually see in the water areas on either side of the membrane, you will see the hydrophobics on the inside and the hydrophilic amino acids on the outside. But inside that cell membrane where there's no water, it'll be opposite. Interesting. So it'll just flip in. So it depends on where it's located, how this thing's shaped. So you can start to see how these properties of the R group, of the side chains of the protein, of the amino acids, are going to start to make this ball up in a certain way. And depending on how that structure is, you'll have a function from that, okay? And so that's the tertiary structure. Now, of course, also there's other things like van der Waals forces. If you don't know what van der Waals forces is, it's basically a shifting of charge. So it's where you have kind of a polar molecule that kind of has some charging, but in a certain condition, and I'm not going to explain it very well right now. It takes a long time to explain, but in a certain conditions, you'll have a shifting of the charge, which will create kind of like a force, kind of like a force that'll help them to come together or be repelled from each other. And so that's tertiary structure. And then quaternary structure tertiary structure is the one you're going to be tested over the most in AP biology because it's the one that affects the function of the protein. Okay. Quaternary structure, you're not going to see quaternary in all proteins. Quaternary structure is whenever you have more than one protein that's folded tertiary style locked together. So this is when, whenever we did our blast lab, we did hemoglobin subunit A. Okay, well then there's a subunit B. There's actually multiple subunits of hemoglobin. When those subunits come together and they are locked together with bonding, just whatever type is available, then they will create a large globular protein called a quaternary structure protein. But not all proteins make quaternary structure. In fact, a lot of proteins just stop at tertiary. Pretty much all of them have a tertiary structure because that's what drives their function. So as long as they have that, then they're in place. Now, I briefly mentioned if you have a mutation. So consider if you have a mutation in your DNA, it would go through all the coding process, but it would have the wrong in the end. It could have, in the end. So here's the codon chart. Look at the codon chart. Look at how you can have multiple amino acids coding for the same, multiple codons, coding for the same amino acid. So if you have a situation where you have a codon, typically it's that third letter, that third base, third nucleotide in the codon. If that switches, a lot of times it won't make a difference. So if you have a weird little mutation that just codes for that third letter, then typically you're going to probably even call for the exact same amino acid. That's a silent mutation. It doesn't really affect anything. Silent mutations can also be because even though it affects something, maybe it calls for another hydrophobic one. And it was a hydrophobic one to start with. And it's a different amino acid, but it has the same properties. And so for the most part, It doesn't really change much. However, you can get a deletion. And if you get a deletion of a DNA base, a DNA nucleotide, if you get a deletion in your mRNA and all of a sudden there's something missing that should be there, we call that a frame shift because it shifts your whole reading frame of your mRNA. Because remember, we're reading in threes. And so now all of a sudden, oh gosh, what should have been three, now one of them's missing. And so it grabs the first nucleotide from the next one to complete the codon, and then domino effect. And those are sometimes a huge mess. They can end up in what we call a nonsense mutation. A nonsense mutation is like where it calls for a stop codon, or a stop terminal. It's a terminal codon. Pardon me. It's a terminal codon way before it should be. And so it's like you just have a little chunk of your protein, and then it was like, oh, time for the release factor to come in. That's bad. That's called a nonsense mutation because it literally makes no sense. A missense mutation is one where you may have a little difference in your mRNA, but it's not enough to really make a big difference. It could make a big difference, but it's not like stopping the whole entire protein. Like there's still a protein there. It's just not really doing what it should be doing. Okay. A lot of our genetic disorders are because of missense mutations, nonsense mutations, little proteins that just don't work because of some mutation in the DNA that caused them to have a misshapen protein. And so basically that's your frame shifts are usually some of the worst ones because your frame shift changes the whole rest of the protein depending on where it falls. Now, if it frame shifts at the very end, sometimes it's not that big of a deal, but that's because whenever it gets to its tertiary structure and the R groups are supposed to interact in a certain way to form a certain shape so that the protein can do its thing, it doesn't work anymore. And then your protein. So cystic fibrosis, wherever you have a buildup of mucus in your lungs, misshapen protein doesn't work. It's a channel protein, so it's not getting rid of some of the stuff it should be. Tay-Sachs, we watched a video of it in my class. It's so sad. Fat builds up in your brain, and babies usually die by the time they're 3 or 4 because they can't get rid of the fat in their brain. One messed up nucleotide, and the protein's misshapen and doesn't work. That's supposed to get rid of the fat in your brain. So some of our genetic disorders are really, really just the result of a small mutation in DNA that's inherited. But anyway, so hopefully you understand how protein folding works and how the different bonding works. I recommend watching this video at least twice because it's kind of a lot. And this is actually really important to understand how proteins fold and do their job in the cell. Okay, I hope that helps. Make sure you ask me any questions if you have any. I will see you soon.